So I'm going to open, and Sarah is going to continue. Welcome everyone to the Developmental and Pediatric Cell Atlas meeting. Sarah and I are going to open with a brief overview, not just generally of the Human Cell Atlas, but specifically of the developmental and pediatric activities in this context. So next slide, please. So the biological network in the Human Cell Atlas that focuses on the Developmental and Pediatric Cell Atlas focuses on really energizing and bringing together multiple communities that want to catalyze progress on these specific areas. And this includes everyone from developmental biologists and embryologists to clinicians, those focused on pediatrics, as well as on other on specific diseases, and technology experts, computational biologists, and ethicists. Next slide. As you know, the HCA has been progressing really nicely, studying 18 organ systems, and from our last cellular census, 46 million cells and counting. And next slide, please. Within this context, there were studies from many individuals, almost 7,000 of them, and almost 8,600 samples. And next slide, next click. Studies from human development have been really one of the areas of great advance. By now, almost 6 million cells from almost 200 individuals and almost 900 samples. Next slide. Just to put it in context, the developmental network is in the context of many other, the developmental and the pediatric networks are in the context of other networks. What's unusual is that for pretty much any other network, the focus is a particular organ and system, whereas here we have to take a cross-system view. Next slide. The network is led by Maz, Hanifa, Stan Linerson, and Deanne Taylor. You can contact them in development at humancellatlas.org. You should join HCA if you haven't already, again, on the website. In the developmental network, there are 78 members to date. The pediatric network is expanding, and please join today. Next slide. So today we're going to take you through several items. We're going to start with a brief overview of the roadmap for the developmental atlas, give a glimpse into the developmental atlases that have been collected today. And then I'm going to switch to Sarah, who is going to talk on the role of developmental atlases used as a reference for disease, both pediatric diseases as well as adult diseases. A little bit on the unique role that model systems play, including model organisms and organoids in the context of understanding development and regenerative medicine in this context, as well as we will close with the role of partnership across programs. Next slide. So starting with the roadmap, actually very recently, Maz, Hanifa, and Maz, Stan, Deanne, and many of our colleagues have published a new roadmap as a piece in Nature that highlights the key features needed for atlases of human development and pediatrics, the need for cross-disciplinary teams, the need for samples that come both from human individuals, model organisms, organoids, and cellular systems, the technologies that need to be used in not just single-cell genomics and spatial genomics, but also imaging techniques and the central role of computational inference, the different levels of resolution at which we do our measurements, and finally, the need for approaches to integrate the atlas and serve it to the community. Next slide. There are unique features on how to construct a developmental and pediatric atlas, so I'll focus on those. And there are three major aspects that are distinct when we think about this. The first is that developmental biology is highly complex and involves protracted timelines, and we have to think about this dynamic at multiple timescales. The second is that the process of cell type specification is intertwined with regulatory events that are not cell autonomous, but happen in a spatial-temporal way. So it depends on where the cell is positioned and what are the cells around it and what are the signals that it received. And so we have to understand these features. And finally, the external signals are not just molecular, they involve signals such as molecular forces, as well as the onset of cell type specific genetic programs driven by transcription factors controlled by upstream signaling pathways. And so all of these features generate unique challenges. The second is that to really understand development, we have to understand things in 3Ds. And that includes things both in the embryonic and in the fetal period. that we then can assess using different types of techniques, but most of these techniques themselves do not perform their measurements in CD, and we have to assemble things back in the end into the 3D context. Next slide, please. And finally, that there's a set of queries that we want to understand in the context of a developmental atlas about what coordinates organ development across the whole embryo. And this is always a temporal query that looks at the difference between different time points. So we want to understand how cellular composition changes dynamically, how cellular interactions change dynamically, what are the changes in molecular mechanisms at many different layers. Here we highlight the chromatin. What are the cellular programs that change, and what are the multicellular structures that change. At all of these levels, there are also applications that are translational. Sarah is going to come back to that, both in designing cells for regenerative medicine, in tissue engineering, and also in identifying molecular therapeutic targets that can be used both in the context of pediatric diseases and adult diseases that recapitulate development, such as cancer. Next slide. To put it in a broader context of the atlas, in the general HCA atlas, many of these questions come to be. And for those who were at the general meeting, we really highlighted how systematic querying of the atlas using computational means is really playing a central role. Next slide. There's different types of technical approaches that one needs for this process. For example, clustering plays a key role in single cell data analysis. And here we're highlighting RCA2, an algorithm that combines reference projection with graph-based clustering. And that allows us to relate information from different levels. Next slide, please. For spatial analysis, cell segmentation is essential. And so SSAM is a cell segmentation-free computational, cell segmentation often plays a critical role, but cell segmentation is also a challenging aspect. And SSAM is an approach that allows us to analyze spatial data in a cell segmentation-free way. Next slide, please. We also want to connect cell lineage, fate, and state, especially in human, for model organisms that Sarah is going to come back for. There are going to be many different approaches, and all of these are rely on engineered tracers. In human, we have the challenge that we cannot apply these engineering methods, and we really have to find things that we can sequence straight from the cell. So single cell DNA sequencing is expensive, but gives us the richest information, and mitochondrial DNA tracing is a cheaper approach, but gives us slightly lower resolution data. Next slide, please. And so to give you a glimpse of the developmental atlases that are being assembled, now there has been tremendous progress over the last couple of years. Next slide, please. And that will give you also a sense of what to expect over the next 3 days. And so in the program, we will hear about developmental and pediatric atlases, about model organisms and organoids in the context of regenerative biology, applications of development and application of relevant technologies. And we will have breakout sessions both to discuss specific atlases, but also we want to highlight in particular the ethics section, because studies in this context require unique ethical considerations. Next slide, please. And so this will be a very brief series of vignettes. Atlases focus both on very particular organs and on a broader view. So here, for example, is an atlas of the corneal cell types that starts at 10 days PCW and goes all the way to adulthood. The atlas of the adult human cornea in conjunctiva reveals a signature of various cell subsets, but then doing single cell analysis of the human developing cornea shows state-specific, how the cells are defined at specific stages, and highlights that both for epithelial, stromal, and endothelial cells, as well as allows identifying unique signatures for specific cell subsets. Next slide. A recent developmental skin cell atlas looked both at the developing skin and at the healthy adult skin. It visualized the cells at each of these features and found how what are the characteristic proportions of the cell states at different time points. Next slide. An atlas of the gut again, characterized it both at the embryonic and the fetal and the pediatric stages. There are adult atlases of the gut already, and again, show the changes in cell abundances at different developmental time points. And Sarah will come back to many of these atlases and show specific vignettes that connect to disease. Next slide. An atlas of the thymus, again, can show the changes in the cellular composition, the development of specific stromal cell populations in the human fetal thymus, and how those vary across the human groups. And the next one, sorry. There's an emergence now of atlases that are starting to look at other layers in an attempt to understand the processes rather than only characterize the cells as they are, and profiling of chromatin plays a particular central role in this context. And so a beautiful recent study, previous slide. So a beautiful recent study used a very high throughput single cell chromatin profiling SkyAttack to profile chromatin accessibility from 800,000 cells across 15 fetal organs. And that allowed, for example, to identify transcription factor motifs that are found in the accessible site of each cell and find the ones that try to explain the best cell type association. And by this, find regulators of the developmental process. Next slide. Because of the need to relate these findings at the mechanistic level, there is particular importance of connecting between human atlases and fetal atlases. And so, for example, in the mouse, between human atlases and mouse atlases. And so in the mouse, for example, we might have access to more developmental stages or can do genetic manipulations that, of course, will not ever be possible in the human. And so projecting human and mouse embryonic trajectories and relating their expression can identify both conserved and divergent patterns and understand interpolational mechanisms. Next slide. And so here I'm going to turn to Sarah, and she's going to take us through the role of these atlases as a reference for disease.
Thank you, Aviv, and a very warm welcome to everybody here. I know there are hundreds of you out there, and it's such a fantastic and exciting point in the history of HCA to be talking to you today at this very first developmental and pediatric focused meeting. And of course, for me especially, this is a personal pleasure because I started work on human cell atlasing by reconstructing the placenta, which is the organ that supports the whole of pregnancy in embryonic and fetal development. And so being together today is really special, and I'm so grateful to the whole program committee, Gary and colleagues and the funders that they've made this possible. And thank you for Aviv for laying out that vision for how we're going to move forward as a community to build the atlases of human development over time. And we hope that this also catalyzes the postnatal and pediatric communities to crystallize and energize the communities to come together. So in the white paper, we laid out the different translational applications that an adult cell atlas can have. And this is actually equally true for human development, and possibly even more so from many points of view that I'll lay out in the next few minutes. And we have breakout sessions 1 and 2 in this meeting dedicated to that. And one example is cancers, pediatric cancers, which are often very different from adult tumors. And even that is exemplified in glioblastoma, shown in this nice publication that compares pediatric and adult glioblastoma, which is a terrible tumor. And in this paper, basically from Sam Bishati and collaborators in my department and elsewhere, where we're showing that the markers for neuroblastomas, which are neuroectoderm derived on the fetal, have a signature related to fetal adrenal medullary cells. And we can use the information comparing adult GTEx, the tumor, and fetal targets to identify markers that would be suitable for disease and not have off-target effects. And so that sort of really powerfully illustrates an application of studying human development and comparing it to cancer and adult tissues. We also have an example here, and this is work from my group, Kirsten Meyer, Marko Nikolic, and others on medRxiv actually, where we're looking at pediatric healthy airway samples and blood, and then also comparing that to adult samples, both in the healthy reference and in disease. And what's amazing here, so this is children, kids, not embryonic or fetal samples, or postnatal, and understanding the difference of these kids compared to adults in the airways and the blood is actually, tells us about how important that stage is, both in the immune system and the epithelium. And
Sarah, we're at time.
There's a little bit of echo again from somebody. And this beautiful paper, again, from members of the Human Cells Community, Roland Isles and collaborators, again echoes that point about the difference between pediatric and adult, and shows how the kids have a more active early innate immune response in terms of viral sensing, and that helps them. Understanding that also gives us insight into the mechanism for how they're able to clear the virus more rapidly than adults very often. Again, coming back to the skin that Aviv mentioned earlier, we show here in this beautiful collaboration with Muzz and Fiona Watt's group on atlasing skin in development and adult, that we have a program in the fetus between vascular endothelial cells that are immune active, immune signaling, and talking to macrophages. And that expression program is actually re-hijacked in inflammatory disease. And this is the same sort of paradigm or pattern that we are seeing here in pediatric Crohn's disease from sample biopsies from the terminal ileum, and comparing them to intestinal development during embryonic and fetal stages, and showing that there's a program of expression linked to proliferation and inflammation that's again hijacked and reused by the inflamed cells, and that's in the epithelium, whereas we're seeing the same sort of pattern here in this work that's on bioRxiv and in Press in Nature, where we've studied the developmental stages of the intestine and in the adult, all the way through the different regions of the gut, from the beginning of the small intestine down to the rectum. And what we're seeing here is this gut-associated lymphoid tissue formation that you can see in these gray dots here that are sitting beneath particular epithelial cells, and using factor analysis on the computational integration of the single cell and spatial data, we're showing that the mesenchymal cells, mesenchymal lymphoid tissue organizer cells, these very specialized cells in this gut-associated lymphoid tissue are signaling to the other immune cells and calling them in. And this expression program, again, I'm not showing it on this slide, but it's again hijacked and leveraged in the ectopic lymphoid tissue formation in disease. And so this is again showing that understanding human development and the mechanisms in human development can tell us about the mechanisms of formation of these tissues in disease, where we often just have a snapshot, whereas in development we can get these different time points and see the genesis of the tissues. Using the data from pediatric IBD, from rare disease, as well as the more complex common disease forms, we are able to map these IBD pediatric forms in terms of enriched cell types, and some of them are shared between the different genetic categories of disease, and some are specific. And this is a sort of review work that's unpublished. So really the point that I want to make in this very important sort of summary and overview is that human development is incredibly informative for both pediatric but also adult diseases, and pediatric atlasing and pediatric understanding how children work can actually is very different and can be really informative for adult disease. So I'm going to move on to not human cell atlas, but other model systems for development, and they include cells in culture, and of course the early pre-implantation embryo can be studied very effectively in culture, because that's prior to implantation into the uterus. I mentioned the placenta before. This is prior to that stage. And of course, research on these culture systems is permitted from an ethical point of view, and allows us to mimic and also intervene even this famous CRISPR experiments by Kathy Nayakan and so on. We'll discuss the ethics around that and interventions in tackling later stages, and I invite you to join the breakout session. I think this is a really important topic because the later stages can, of course, only then be mimicked in the dish using sort of increasingly complex co-culture systems, which are super cool and are very exciting and sophisticated, but aren't quite the same as the in vivo at this point. And of course, we have a traditional set of model organisms to study human development, the famous fly, with the early embryonic patterning being discovered by Nusslein, Vollard, and Wieschaus. And the model organisms are incredibly powerful. You know, the different organisms have been leveraged for studying different aspects of development, different purposes, but their recapitulation of human physiology is variable, I would say. And this is the size of the dots is summarizing that. And of course, the genetic manipulation is the big benefit of them. And that's where the cell culture and organoid systems come in, is that they can recapitulate human physiology very, very effectively, at least for certain small tissue units or cellular units. And so we've learned a huge amount from model organisms, including mice here, also early stages that aren't accessible, as I've just discussed, in terms of the early development of visceral endoderm cells, or the early stages, and the patterning, where you've got even prior to organ definition. And we're now getting to the stage where the mouse brain is a kind of beloved model here, the development of the mouse brain for the human cellulose community. It's been used by a lot of members of the community also as a way of test driving computational and experimental methods. And what you can see here is spatial analysis of the developing mouse brain and integrating spatial transcriptomics and RNA sequencing with Tangram. There are other methods out there, and as well for this kind of thing. And these methods are absolutely at the heart of what we can do in terms of a detailed reconstruction in 2D and 3D. And the models are extremely informative and kind of often provide an anchor when we get into human systems. And the same is true here for this comprehensive atlas of the mouse brain, starting much earlier than we can study human, telling us about spatial organization of progenitors, and again, a tour de force, and very exciting to see this analysis of model organisms.
Sarah,
and of course, the challenge in human is, as I mentioned before, the lack of the...
Sarah, sorry to interrupt. We just wanted to do a time check.
I'll accelerate.
Okay.
In the model organisms, we can't CRISPR them, which you can get at the lineage. Very difficult in human. Regenerative medicine is the topic of this meeting, Gary, as I know, and you want us to emphasize that the value of the developmental cell atlas. Of course, children heal better than adults, and even in the embryo and fetus, we have fantastic regenerative processes. And so we want to emphasize that funding development in pediatrics will tell us about these processes. We have different systems, obviously, ESC, iPSC derived, and we'll hear about, we'll have a keynote also that will relate to modeling disease cell therapy and efficacy and model organisms. I think model organisms will be more Kat's emphasis. I'll go very fast now, but just to say that there are systems of transcription factor overexpression and modeling of screening of transcription factors for inducing different cell lineages. And these cell-based and organoid systems will allow us to gain mechanistic insights, also can function as platforms for toxicity and efficacy of drugs, and eventually actually be a therapeutic substrates and products themselves. And of course, the human cell atlas, the pediatric cell atlas, and development cell atlas provide a way of QCing these in vitro systems, and also provide us with the ingredients to program them as we've described in this review recently from the Organoid Cell Atlas Working Group, from Christoph and Barbara and others. And we look forward to Hans and Barbara's talks that will elucidate these topics. And this is basically just work from us that I mentioned earlier, that there are many other works that show that you can generate multiple lineages in parallel in organoids. iPS-derived systems have been favorites for the brain, and this shows basically how single-cell RNA sequencing can tell us about what is in the dish in these brain systems, compare them and quality control them. And we look forward to hearing talks on this topic in this meeting. And I just want to finish with a big thank you again, Gary, Diane, Bruce, Sten, all the other members of the committee. Aviv and I obviously chimed in, and the supporters, the University of Toronto Medicine by Design, and all the other funders who've made this possible. An exciting time, and welcome everybody, and thank you.